Hi everyone. My project focuses on white matter microstructure and Alzheimer's disease pathology in the brain. We all know that Alzheimer's is characterized by memory loss and cognitive dysfunction, but this is really the late stage of the disease. And before that, there's a very long preclinical stage during which brain changes are already occurring. They start with the accumulation of amyloid beta plaques, followed by tau proteins, and then leading to neuronal loss. But how white matter alterations might fit in this picture is still not clear. And the starting point from this project was really based on the spatial pattern of amyloid and tau deposition. Early on, tau is very circumscribed. It's present in the medial temporal lobe, whereas amyloid is more widespread in medial regions and reaching the cingulate cortex, temporal cortex very early on. And when we look at this picture, we can see that the regions where pathology accumulates are connected by very well-defined anatomical bundles. For example, the fornix is the key bundle that originates from the hippocampus. Looking on the amyloid side, we can see how the cingulum bundle is connecting the key early amyloid regions. And so the objective for this project was to investigate the associations between amyloid and tau and the white matter microstructure in specific bundles that are connecting the brain regions where Alzheimer's pathology is found. To do that, we turn to the PREVENT-AD cohort. This is an ongoing study at the Douglas where we enroll cognitively normal older adults who had higher risk of Alzheimer's disease because they have a family history of the disease. All participants underwent diffusion MRI. This is the MRI modality used to be able to reconstruct the white matter architecture, yielding these beautiful tractograms. All participants also underwent positron emission tomography to be able to image the presence of amyloid and tau in the brain. From our tractograms, we then extracted our bundles of interest. They were the uncinate fasciculus, the fornix, and then the anterior and posterior part of the cingulum. And in each bundle, we then extract typical diffusion measures that mainly represent the amount of diffusion that's possible along those bundles. And then as a last step, we look at associations between the different diffusion measures and the amount of amyloid and tau directly at the endpoints of the bundles. I just want to say this project and the methods used was done in collaboration with the lab of Maxime Descoteaux at Sherbrooke University, so I'm very grateful for that collaboration. Turning to the results, we see associations in 3 of our 4 bundles of interest. And starting in the fornix and the anterior cingulum, we see associations only with amyloid. And the pattern that we see is that there is higher anisotropy and less diffusivity with the presence of amyloid in the brain. Basically, what this means is that there is less space for the water molecule to diffuse along this bundle if there is more amyloid in the brain. Turning to the uncinate fasciculus, this is where we saw the strongest associations. They were present both with amyloid and with tau. And the key thing is really that the directions of association, that the pattern that we see is very similar as in the 2 other bundles. So again, here, less diffusivity, less space for our water molecules to move along these bundles is associated with higher amounts of pathology in the brain. Taken together, if we bring back our little timeline of Alzheimer's disease, what we see consistently across 3 bundles is higher anisotropy and less diffusivity. However, this is in contrast with what is observed in the late dementia stages, where we see a typical pattern of white matter neurodegeneration with increased diffusivity in the brain. So I think this highlights that there is possibly different mechanisms at play between the early stage of the disease and the late stages. And focusing specifically on this preclinical population can thus help us understand better the early pathogenesis of the disease. And with that, I'd like to thank you for watching. If you have questions or want to discuss this project more, don't hesitate to reach out. And I hope you're all having a great summer. Thank you.